Good morning, students. Here I am back with Module 2 of Chapter Nutrition in Plants for Class 7. But before starting Module 2, let's recall and recapitulate the content done in previous class of Module 1. In Module 1, we have studied about autotrophs. All green plants are autotrophs and are self-sufficient to produce their food by themselves. Autotrophs prepare their food by the process of photosynthesis. Plants require raw materials like carbon dioxide, water, sunlight, and have chlorophyll in them, which are essential to carry on the process of photosynthesis. The products of photosynthesis are food in the form of glucose and oxygen, which is an essential component for the survival of all living organisms on the earth. As you know, students, the process of photosynthesis is a very important biochemical process carried by green plants to produce food and oxygen. But the process of photosynthesis cannot be carried by the plants if any one of the raw materials required for photosynthesis is missing or absent. So, in this module, we will study and demonstrate few activities to prove that all the raw materials are essential for photosynthesis. Here in this module, we will perform some activities to demonstrate that starch is formed during photosynthesis, to demonstrate that chlorophyll is necessary for photosynthesis, to demonstrate that sunlight is necessary for photosynthesis, and to demonstrate that carbon dioxide is necessary for photosynthesis. Let's start with activity 1. To demonstrate that starch is formed during the process of photosynthesis. The materials required for this activity are leaf, boiling water, alcohol, iodine, test tube, spirit lamp, dropper. Now, what is the procedure? Take 2 green potted plants and label them A and B. Put them in dark for 48 hours. Let plant A be kept in dark place and plant B in sunlight for at least 4 to 5 hours. Pluck 1 leaf from plant A and 1 from plant B. Boil the 2 leaves for 2 to 3 minutes in water. Boiling of leaf in water will kill the leaf, disrupt the cell membrane, and soften the cell wall, allowing iodine to enter the cell. Now, put the leaves in test tube having ethyl alcohol and keep the test tubes in beaker containing water. Heat it till alcohol in the test tube becomes warm. Please make a note, students, that the test tube with alcohol and dipped leaf in it should not be heated directly. It should be kept inside a beaker with water in it. Take out the leaves from alcohol, wash them in warm water and place them in a glass plate. Now, when all the chlorophyll from the boiled leaf has come out into the alcohol, the leaf is left colorless. This is done to remove chlorophyll and to make the leaf colorless so that change in color is noticeable. Put a few drops of iodine solution on each leaf. Now, what do you observe? Leaf B turns blue-black, but leaf A does not. Conclusion is, plant B has starch, but plant A do not have starch. Photosynthesis occurred in plant B, but it was kept in sunlight, but not in A as it was kept in dark. This shows that starch is formed during photosynthesis. Now let's come to activity 2. Here we will study and demonstrate that chlorophyll is necessary for photosynthesis. The materials required for this activity are Coleus plant or Croton, alcohol, test tube, beaker, iodine solution, white paper, burner, tripod stand with wire gauge, faucet and dropper. Students, Coleus plant or Crotons have variegated leaves. On certain plants like Crotons, the leaves are spotted white, yellow or red with green. Green part of the leaf contains chlorophyll and non-green parts do not have chlorophyll. Now, keep it in the sunlight for a few hours and pluck a leaf. Sketch its shape on a plain white sheet. Mark the green and non-green areas of the leaf on the white paper. Carry out the test for the presence of starch as you have done in the previous activity. What do you notice? The leaf marked green in the sketch shows blue-black color with iodine. The non-green area remains unchanged. So, the conclusion is that starch is present in the green area of the leaf where chlorophyll is present and photosynthesis has occurred. So, we can conclude that chlorophyll is essential for photosynthesis. Now, let's discuss about activity 3. Here, we will demonstrate that sunlight is necessary for photosynthesis. What is the procedure? Keep a green potted plant in dark for about 48 hours to destarch its leaves. What is destarching of leaf? A green plant synthesizes food during daytime and leaves are filled with starch. During night, starch moves from leaves to various other parts of plant. Before the next day, leaves are left starch-free. Now, pluck one leaf and test it for iodine to confirm that the plant is destarched. Cover a part of healthy leaf on both the sides with a strip of black paper. Place this plant in sunlight for at least 4 hours or more. Pluck the leaf and remove the black strip. Boil the leaf first in water, then in alcohol. Wash the leaf with warm water and put it in a petri dish. Pour a few drops of iodine solution on the leaf to test the starch. What do you observe? The uncovered part of the leaf that was exposed to sunlight turns blue-black, while the part of the leaf which was covered with black strip does not turn blue-black. This shows that starch is formed only in those parts which are exposed to sunlight. No starch is formed in the covered part which could not get sunlight. Hence, sunlight is necessary for photosynthesis. Coming to activity 4. Here we will demonstrate that carbon dioxide is necessary for photosynthesis. What we have to do? Keep a green potted plant in dark for 48 hours to destarch its leaves. In a white mouthed bottle fitted with a split cork, take about 2 to 4 ml of potassium hydroxide solution. This solution is used to absorb carbon dioxide if present in the bottle. Select a healthy leaf from the plant and insert half of it in the bottle between two pieces of the cork. Place the setup in sunlight for at least 4 hours. After 4 hours, remove the leaf from the plant and out of the bottle. Test for the presence of starch by boiling the leaf in water, followed by alcohol, and then use iodine solution. Now what do you find? The part of leaf outside the bottle turns blue-black, but the part inside the bottle remains yellowish. So here this shows that photosynthesis had taken place in that part of leaf which was outside the bottle and received carbon dioxide. The part of the leaf inside the bottle did not receive carbon dioxide, so no starch was formed. With this, I end up module 2. Thank you, students. Have a nice day.